When someone has a genetic test for a rare condition, their sample may be sent to our Rare and Inherited Disease Laboratory for analysis. Lab technologists will extract the DNA from the sample. Using specific chemical mixtures and machines to speed up the process, they break open each cell and isolate the DNA. For a sequencing machine to read DNA, it must be turned into something that is known as a library, which requires the DNA to be separated into smaller chunks or fragments that are easier to read. For many rare disease tests, instead of looking at all of the DNA, which is more expensive, scientists will look at just a small number of genes, called a panel, that are known to cause specific conditions or diseases. This means they only separate out the fragments of DNA that they are interested in. There are lots of different panels, like ones for epilepsy or metabolic disorders. The library then goes into a large sequencing machine in the laboratory. These machines read each letter or base of the genes. The end result is a lot of data, a long list of all of the bases that make up each gene. Bioinformaticians have developed tools that help computers read all of that data and then produce a document that highlights changes in the genes, also known as mutations. That document is then sent to our team of clinical scientists. They use all of their training, genomics knowledge, and clinical information to interpret what the gene changes mean. They work to understand which of the changes may be causing the symptoms a person is experiencing. Once the clinical scientists have worked all of that information out, they generate a report for clinicians so they can give results back to patients and their families.